All right, today we're jumping into one of the most fundamental processes in all of life, glycolysis. Think of it as the starting engine that powers pretty much every living thing. So let's break it down step by step. Every single living cell, from a tiny bacterium to the neurons firing in your brain right now, needs energy to do its job. And their go-to fuel source? It's usually a simple sugar, glucose. So the big question is, how do cells actually get the energy that's locked inside that molecule? Well, the answer starts with glycolysis. This is the very first step of cellular respiration. And what's really cool about it are two things. First, it's anaerobic, meaning it doesn't need any oxygen. This is a huge clue that it's an incredibly ancient pathway, probably evolving way back before Earth's atmosphere even had much oxygen. And second, it all goes down right in the cell's cytoplasm, which makes it this universal baseline way for pretty much all cells to get a quick energy boost. So at its heart, the whole process is a really neat bit of chemical math. You take one molecule of glucose, that's our 6-carbon sugar, and you split it perfectly in half. What you end up with are 2 molecules of pyruvate, and each of those has 3 carbons. It's that split right there that kicks off the whole energy release. But you know how they say you have to spend money to make money? Well, it's the exact same idea for energy inside a cell. Glycolysis doesn't just start making energy out of thin air. It kicks off with a phase that's perfectly named the energy investment phase. Okay, so to get the ball rolling, the cell has to spend 2 molecules of ATP. First, an ATP gives a phosphate to glucose, turning it into glucose 6-phosphate. After a little shuffling around, a second ATP comes in and adds another phosphate. Now we've got fructose 1,6-bisphosphate. This molecule is kind of unstable. And because of that, it immediately splits into 2 separate 3-carbon sugars, getting them perfectly prepped and ready for the next phase. So, the key takeaway from this first part is the cost. The cell is now down to ATP. Just keep that number in your head because the payoff is right around the corner. And here we go. Welcome to phase 2, the energy payoff phase. This is where that initial investment really starts to pay dividends. All right, now the good stuff starts. Each of those 3 carbon molecules gets oxidized. This process passes electrons to a carrier molecule called NAD+, which creates the energy-packed NADH. Then, in the final steps of the process, some high-energy phosphate groups get handed off to ADP, and bam, we get our target, ATP. And remember, this whole thing happens twice, once for each of those 3 carbon molecules we made earlier. So, because that whole payoff phase runs in duplicate, one for each of the 3 carbon molecules, we get a pretty nice return on our investment. When you add it all up, we've generated a total of 4 ATP molecules and 2 NADH molecules. Okay, so we've seen the investment and we've seen the payoff. But what's the final tally? What's the net result? And why is this so important? Let's look at the final numbers. We put in 1 glucose molecule and invested 2 ATP. What did we get out? 2 pyruvate molecules, 4 ATP, and 2 NADH. So when you do the math, we spent 2 ATP but made 4. That means the net profit for the cell is 2 ATP and 2 NADH. Now, that might not sound like a huge amount, but this is incredibly fast, easy to access energy, and it's absolutely vital for tons of things the cell needs to do. But that leaves us with a big question, right? We've got our ATP, our NADH, and these 2 pyruvate molecules. The story's not over yet, so what happens to pyruvate now? Well, its fate really depends on one thing. Is there oxygen around? If there is, what we call aerobic conditions, then that pyruvate gets shipped out of the cytoplasm and straight into the cell's power plants, the mitochondria. And once it's inside the mitochondria, pyruvate becomes the fuel for the next big stages of cellular respiration, the Krebs cycle and oxidative phosphorylation. These are the processes that will completely break it down and generate a ton more ATP. I mean, way, way more than glycolysis could ever make on its own. Okay, so this is a classic biochem pathway. But how does it connect to the world of bioinformatics? Where's the link? Think of glycolysis as just one small road on a massive metabolic superhighway. In bioinformatics, we use amazing databases like KEGG to map out these incredibly complex networks. This lets researchers do some really cool stuff, like model what would happen to the whole system if one gene for an enzyme has a mutation, Or they can even compare the metabolic efficiency of different organisms. And they can do it all from a computer. But in the end, it all comes back to this one central idea. It doesn't matter if you're analyzing an entire genome or just looking at a single cell, glycolysis is and always will be the absolute foundation of how cells produce energy. So there you have it. From one glucose molecule to two pyruvates, it's this intricate 10-step dance that kicks off the flow of energy powering all life as we know it. Understanding it is just fundamental to both biology and bioinformatics. If you want more clear scientific explanations like this one, make sure you subscribe to BioInfo Bytes.